observation from the population. And so this has mostly been an observation in quantitative genetics, and in population genetics, mostly we've found evidence for forces that remove variation instead of maintain it. So again, directional or purifying selection instead of things like balancing selection. So obviously there's a bit of a disconnect between the expectations from quantitative and population genetics, and it's unclear how to reconcile that. So this is just one hypothesis that we had, was that it's possible that the lack of evidence for maintenance of variation in populations could just be an artifact of technical approaches. So we decided to try deep coverage of a single population to see if we could find more evidence for heritable variation than in other panels. So we generated 170, well, it was actually like 200 originally, isofemale lines, and inbred them with full SIB mating for 15 generations. And we got them from a single, large, stable population in Zuma Beach. And when I say stable, I mean, for example, in Southern California, there's no yearly population crashes during the winter or anything like that. There's consistent resource availability. So there's going to be that kind of seasonal cycling of population sizes. So we called SNPs according to G8K best practices. Just use standard filters like masking heterozygous bases, biallelic loci, and then ended up with 6 million SNPs to work with. So I'll take a moment to explain what we're looking at here. So this is a graph of pi, theta, and Tajima's D, and on the right-hand axis is pi and theta in blue and gold, and on the left-hand axis, is Tajima's D in pink, and there's a pink line at 0. So the values of pi are consistent with other studies that have measured it in Drosophila simulans, like the Gunn study in 2007. However, as you can see, we found pervasive values of positive Tajima's D in regions of normal recombination. So you can see the pink line is above 0 pretty much everywhere. And we were really surprised by this result, right? This was not what we were expecting. Usually people find evidence for lots of negative Tajima's D, or so we weren't quite sure what to make of this at first. We did find a study where they found a similar result. It wasn't quite as positive, but it was pervasive positive values of Tajima's D in a 2015 paper. They didn't really go into why it might have been positive, and just, it was 84 lines in 5 populations, so slightly different makeup of the panel as well. So in order to try and make sure that we weren't making some kind of mistake, we applied a bunch of sensitivity analyses. So we looked at different quality filters, including different types of SNP calling. We excluded individuals with lower levels of inbreeding, or isofemale lines with lower level of inbreeding. We looked for a relationship between heterozygosity and quality of variance. And we also just tried calling these statistics with several different methods to check for computational errors. And nothing altered the patterns that we found of pervasive positive Tajima's D. So there are a couple of explanations for this kind of pattern. There could be demographic explanations, like population contraction or admixture. And there could be biological explanations, like balancing selection or incomplete or soft selective sweeps. So I'm going to go through each of these possible explanations and talk about how our data is or is not consistent with those explanations. So for population contraction, that would be a demographic explanation. You would expect to not see large differences between low and high recombining regions of the genome. And you can see at the origin and end of the graphs, there's negative Tajima's D values. That's the centromeres and telomeres where there's low recombination. Also, our polymorphism estimates are consistent with other studies. So none of that suggests that population contraction is responsible for these patterns. Likewise, if it was something like admixture, you would expect that polymorphisms from different progenitors would be commonly co-inherited, right? So that would manifest as long-range linkage disequilibrium, which we don't observe. Linkage disequilibrium is similar to that calculated in pretty much every panel of Drosophila melanogaster. It's less than 500 base pairs. And you can see that it is a little bit higher on the 4th chromosome, which is expected. There's some recombination on the 4th chromosome in Drosophila simulans, but not as much as on the autosomes. So under balancing selection, you would expect that alleles have a deeper phylogenetic history because they have more time to recombine. So the expectation would be lower LD in regions of high polymorphism, so lower LD in regions of positive Tagina's D. So this is a graph of linkage disequilibrium plotted against Tagina's D. We have two different lines here. So the blue is the line for negative Tagina's D values, and pink is for positive Tagina's D values. In the fourth chromosome, there's only a blue line, which is shown right there. So in the negative region, we see high linkage disequilibrium, which is consistent with expectations. So you would expect that in regions that are undergoing either directional or purifying selection, you're going to have higher LD. Those are also the regions of the genome that have lower recombination. In the positive spectrum of Tagina's D, you actually, if this was balancing selection, you would almost expect the blue line to just continue the way that it's going, right? But what you actually see is a slightly positive slope with Tagina's D. And that's also the regions of normal recombination. So this is not consistent with what you would expect under balancing selection. So the evidence is most consistent with incomplete or soft selective sweeps, where selection is driving alleles to higher frequencies without reaching fixation. So this could be caused by a lot of different things. It could be caused by context-specific fitness, like spatial or temporally varying fitness, or social selection. It could be caused by polygenic adaptation, selection on multiple beneficial alleles at a locus, and it is consistent with the higher LD at higher values of Tagina's D. So just to clarify, we're not implying the super strict definition of soft selective sweeps where, for example, it has to come from something that's been at standing variation and then goes to fixation later, or where there's multiple alleles at one locus that go to fixation. Rather, I think the distinction between the two types of selection are somewhat artificial, and what we're just trying to say here is that instead of the classic hard sweep of one mutation going to fixation, that you're seeing things with softer edges. So for example, in the case of an incomplete sweep, selection could be hard, right? But then it just wouldn't go to fixation. Maybe selection goes in a different direction then. So in order to confirm our results and look at the frequency of hard versus soft sweeps, we employed a haplotype diversity test. So these are the top 10 selected regions on each of these chromosomes, and it's showing the haplotype frequencies in the 1 kb windows. So the first two haplotypes that are the most frequent are shown in blue and dark blue. Gray boxes are singletons, and the more haplotypes you have at higher frequencies, the more of a softer incomplete sweep it is. So higher down at the bottom, if you can see the values, higher H12 values, that's the statistic, means soft or hard selection, and then you calculate a second statistic called H2H1, which distinguishes between soft or incomplete sweeps. So the higher the H2H1, the more likely it's an incomplete or soft sweep instead of a hard sweep. And you can see just from looking at this that on the X chromosome, it looks like there's a little bit more evidence for hard sweeps than on the other chromosomes shown there. So in order to look at basically how pervasive soft or hard sweeps were, we divided up our H12 values into groups. So the top 25% of H12 values is going to be shown in black, the middle 50% in pink, and the lowest 25% in blue. And then within those groups, we're going to plot the density of H2 over H1 values. So, for example, in the black line, we know that those are hard or soft sweeps, but because there's a peak on the X chromosome that is closer to the origin for H2, H1 values, that suggests that harder sweeps are more important on the X. But on the other chromosomes, there's a different story. So H12 values are associated with a range of H12 values, or sorry, high H12 values are associated with a range of H2, H1 values. And there's no peak at lower values. And this supports our theory that most sweeps don't have a hard edge. For example, you can look at 3R, and there's actually a peak at higher H2, H1 values. And for most of them, they sort of crest at more intermediate values. So just to add some support for our claims, we also found evidence for sweeps that other people had found. So like we know our method is basically working in that sense. So for example, SIP6G1 was in one of our top peaks, and that's been known to be involved in a sweep in the Lancaster for a long time. Same with ACE and CHCUF1. Those were both soft sweeps. So in conclusion, many other collections represent many populations. For example, the DGRP was collected at a farmer's market and probably represents a mix of populations. So it's possible that sparse sampling could result in the impression of multiple SNPs at low frequency. That's our hypothesis anyway. And that in reality, there may be actually abundant heritable variation in populations due to context-specific fitness, polygenic selection, and they found evidence for this in other systems as well. So there's been some work recently in C. elegans, bacteria, and rabbits, all looking at the importance of soft or incomplete sweeps. And so this might reconcile these observations from population in quantitative genetics that previously seemed contradictory, that there was this expectation of abundant variation that wasn't being found in population genetic studies. So with that, I'd like to acknowledge all of my co-authors and collaborators on this project and my funding sources, the University of Southern California and the National Institute